Today we'll be talking about cell communication. There are four main processes involved in cell communication: the G protein pathway, the tyrosine kinase pathway, ion channel receptors, and intracellular receptors. The first process I'll be talking about is the G protein pathway. This process is started when a signaling molecule or ligand attaches to a G protein coupled receptor or GPCR. When the ligand attaches to the extracellular side of the receptor activates and therefore changes shape. The receptor's cytoplasmic side then binds to an inactive G protein and activates it as well. The GPCR activates this G protein by causing the GTP, which is similar to ATP, to displace the GDP, which had previously made the G protein inactive. Next, the activated G protein dissociates from the receptor, diffuses along the membrane, and binds to an enzyme which alters its shape and activity. This process is called phosphorylation, or activation of the enzyme by the G protein. Meanwhile, the ligand dissociates from the receptor and frees up the receptor for the next signaling molecule. The changes in the enzyme and G protein are only temporary, however, because the G protein also functions as a GTP-ACE enzyme, which means it hydrolyzes its bound GTP into GDP and P. This makes both the enzyme and the G protein inactive again, so that they can be reused. The GTP-ACE function of the G protein will then turn off or turn on this necessary process in the cytoplasm or nucleus. The next process I'll be talking about is the tyrosine kinase pathway. This process is extremely important in growth and emergency repair. The process starts with individual receptor tyrosine kinases, which are referred to as monomers. These RTKs have an extracellular ligand binding site, an alpha helix, and an intracellular cell tail containing multiple tyrosines. Once ligands bind with the two monomers, the two begin to associate closely with each other as seen in this picture here. They begin to form a complex known as a dimer in a process called dimerization. Dimerization activates the tyrosine kinase region of each monomer. Now that this region is activated, each tyrosine kinase adds a phosphate from an ATP molecule to a tyrosine on the tail of the other monomer, so that the previous 6 ATPs on this dimer become 6 ADPs on this dimer. Now that the receptor is fully activated, it is recognized by specific relay proteins inside the cell. These proteins bind to specific phosphorylated tyrosines. This in turn results in structural change that activates the bound proteins. Each of these activated proteins triggers a transduction pathway, which helps the cell regulate many aspects of cell growth and reproduction. The ability to trigger so many transduction pathways is the key difference between the tyrosine kinase pathway and the G protein pathway. The third process I'm going to talk about is ion channel receptors, aka ligand-gated ion channels. A ligand-gated ion channel is a type of membrane receptor containing a region that can act as a gate when the receptor changes shape. When the ligand binds to the receptor and the gate opens, specific ions can flow through the channel and rapidly change the concentration of that particular ion inside the cell. For example, in the nervous system, neurotransmitter molecules are released at a synapse between two nerve cells. These molecules act as ligands to ion channels on the receiving cell. When the ligand binds, ion flows in, which trigger an electrical signal that propagates down the length of the receiving cell. This is why ion channel receptors are crucial in the nervous system. When the ligand dissociates from the receptor, the gate closes and ions can no longer enter the cell. The fourth process I'm going to talk about is intracellular receptors. These receptor proteins are found in either the cytoplasm or nucleus of target cells. They are mostly for receiving hormones and steroids. The specific example I am going to use will be the steroid hormone aldosterone. First, the steroid hormone aldosterone passes through the plasma membrane here. Aldosterone then binds to a receptor protein in the cytoplasm, thus activating the protein. The hormone receptor complex enters the nucleus and binds to specific genes in the nucleus. Next, the bound protein acts as a transcription factor, which stimulates the transcription of the gene into mRNA. Last, the mRNA is translated into a specific protein outside of the nucleus in the cytoplasm. The final subject I'm going to talk about is the secondary messengers, which are relay molecules within the cell's cytoplasm. These are usually ion-based molecules within a cell. The three most common types are cyclic AMP, or CAMP, calmodulin, or calcium ions, and inositol triphosphate, referred to as IP3. Cyclic AMP, or CAMP, is used in the G protein pathway, which I talked about earlier in the video. CAMP is used to provide cellular responses within a cell. Adenylyl cyclase, which is an enzyme embedded in the plasma membrane, is responsible for converting ATP into CAMP in response to an extracellular signal. This is where the process is started. Once the first messenger or ligand activates the G protein coupled receptor and the GPCR activates the G protein travels across the plasma membrane to phosphorylate adenylyl cyclase. When activated, adenylyl cyclase catalyzes the conversion of ATP to CAMP. The CAMP then acts as a secondary messenger and activates another protein, usually protein kinase A, which then leads to cellular responses. Calcium is even more widely used than CAMP as a second messenger. Increasing the cytosolic concentration of calcium ions causes many responses in animal cells, including muscle cell contraction, secretion of substances, and cell division. Cells use calcium ions as a second messenger in pathways triggered by both GPCRs and RTKs. In this illustration, calcium ions and inositol triphosphates function as second messengers in the G protein pathway. First, the ligand or first messenger binds to the GPCR, which leads to the activation of phospholipase C. Phospholipase C then cleaves a plasma membrane phospholipid called PIP2 into DAG and IP3. IP3 quickly diffuses through the cytosol and binds to an IP3 gated calcium channel in the endoplasmic reticulum membrane, causing it to open. Calcium ions then proceed to flow out of the endoplasmic reticulum and raise the calcium ion level in the cytosol. The calcium ions then activate the next protein in one or more signaling pathways, which cause cellular responses within the cell. Well, this concludes my video. I hope you enjoyed it and learned some things in the process. Have a great day, guys.